Hi everybody. I am really thrilled to talk to you today about the algorithms that let us identify proteins. This is the field that I have been in since 1996. I love it. And I've written a lot of papers about what works well and what doesn't work as well, and it's been a fun place to be. So I hope that today we can take you through everything from the start to the finish of how we get mass spectrometry data that tell us what might be in a sample, work it over with different algorithms until we finally get to a protein list that we can actually trust. So just keep on line here and we will move on ahead. If you're watching this from home, be aware that there's a link below this in the description that gives you the link to the slides directly, so you don't have to watch them here. You can just watch me wave my arms. So on we go. We are going to start by understanding tandem mass spectrometry. Tandem mass spectrometry has been the key method that we use for revealing the sequences of peptides and thus of proteins for a long time now. So we're going to spend a little time talking about how that works. Now, we're next going to talk about comparing MSMS scans. Now, every time I use MSMS, you might be confused by that, but I'm going to just note that's tandem mass spectrometry. How do we compare tandem mass spectrometry scans to peptide sequences that have been drawn from a sequence database? This is, at its nub, the killer application of proteome informatics, the database search engine. From there, we're going to talk about filtering peptide spectrum matches to control the false discovery rate. In other words, how do we prove that the identifications we're looking at are trustworthy to some level? And then finally, how do we infer parsimonious protein lists that can account for all of the peptides that we've identified? So that's kind of the roadmap for today, and we'll just move straight ahead into the first of these. So I'm going to start by thinking that you might have spent a whole lot of time thinking about microarrays or a whole lot of time thinking about next-gen sequencers. And one of the ways in which these things work is through massive parallelization, right? You can measure every probe sequence for a species at the same time in a microarray. You can sequence millions of templates all at the same time in a next-gen sequencer. But mass spectrometry doesn't work that way. Instead of massive parallelization, we're dealing with a high-throughput serial technology, one where our data are captured one at a time. So the mass spectrometer is most generally, in the typical mode of operation, not in some of the newer ones, producing a spectrum of each peptide one at a time, one after each other. Okay, so this is a serial process. Thus, the scan rate, the rate at which a mass spectrometer can produce new tandem mass spectra, in large part reflects the sensitivity that it will be able to achieve. How deep can you go into a proteome and identify new stuff? So, what are the features that all mass spectrometers possess? Number one, an ion source. If it's not an ion, we cannot see it in a mass spectrometer. If you've got something neutral, it's not going to be detected. So we need these ions to carry charge in order to manipulate them, in order to decide which ones stay and which ones go, in order to break them apart, in order to detect them. In a lot of cases, we need to have a charge on that ion. So being able to produce an ion source, having a source in the instrument that can produce these ions matters tremendously. Does anyone know what Nobel Prize was given in the field of proteomics? It was given for ionization, for finding a way to turn things as big as peptides into ions, for two main methods that exist in that space. So next, we have mass analyzer. The mass analyzer is kind of the business end of the mass spectrometer. It's the part that's actually manipulating ions by their mass to charge values. And finally, we have a detector. This detector is going to allow us to say how much did we have at a given mass to charge value. So we have the ability to create the signal, to manipulate it in some way, and then to measure what comes out. Okay, now a key principle that I'm going to return to time and time again is the M over Z value. I realize you might say M over Z here, and that's okay. So the M over Z value reflects the mass divided by the charge of an ion. And this mass to charge ratio is the principle by which almost all ions respond inside a mass spectrometer. Okay, so what does this whole crazy workflow look like? Sometimes it's useful to have just this view from 20,000 feet to get some sense of what's going on. We start with a protein mixture. How complex a protein mixture would you have if you just blew up a bunch of cells? Well, it probably depends what kind of cells, right? So let's say you're blowing up human cells. between the numbers 100, 1,000, 10,000, or 100,000, or a million, which of those is closest to the number of proteins you're likely to find in a given sample? The complete count. Okay, no answers on that one yet, but how about we start with how many genes do we have in a human genome?
22,000?
About 22,000? The number's a little higher than it was maybe last year, but yeah, let's say around 22,000. That's a good count. Now, do we have one protein for every gene? We do not. Sometimes we have multiple transcripts that are possible for a given gene, right? So at present count, if you look at the number of proteins listed in a human proteome database, one you get from UniProt, or one you get from RefSeq, or one you get from Ensemble, you're likely to find a database of around 100,000 different proteins. Now, does every cut of meat you take off of a biopsy contain all 100,000 proteins? No. Some of them are on, some of them are off. Some have produced only one of several isoforms under a given circumstance. So generally speaking, it's probably reasonable to say that detectably a given cut of a human sample is going to have something like 10,000 proteins in it. So does it really make sense in this very first step to digest this mixture of 10,000 proteins to produce 100,000 peptides? Not necessarily. It actually makes the sample a whole lot more complex for you to deal with. But peptides are an awful lot easier to deal with in biochemical separations, and it's a whole lot easier to make sense of the spectra that they produce. So our first step, maybe translating 10,000 proteins into 100,000 peptides, is actually improving our ability to tease information out of that sample. From there, for many samples as complex as a bit of human, we end up using fractionation. Now, fractionation is not used as frequently in South Africa because mass spectrometry feels like a very expensive technology. But in the rest of the world, you frequently find people making use of fractionation, maybe by separating proteins out in a 1D gel, maybe by using reverse phase separation at basic pH, maybe using isoelectric focusing. There are lots of ways you can do this. But that fractionation turns one sample into many. So if you were doing a very deep dive into tissue, you might produce 24 Eppendorf tubes that all contain some fraction of the peptides from that complex mixture. From there, we most typically make use of acidic reversed phase liquid chromatography. So we're going to separate the peptides in time as a function of their hydrophobicity. Now, what makes a peptide hydrophobic? Side chains. That's a very good answer. Yes. So a longer peptide has more side chains, and it has the greater possibility of becoming a very, very hydrophobic molecule. So if, for example, you have a bit of a protein that passes through a transmembrane domain, is it hydrophobic or is it hydrophilic? If it's passing through a lipid bilayer, that part of the protein is what? Oh. Okay, so that's a tough question. All right. So lipids are by nature a very hydrophobic environment, right? You don't find water just passing through the cell membrane, right? So when we have proteins that pass through the cell membrane, they generally have very hydrophobic regions that do so, which means they're going to have a whole lot of isoleucines and valines and leucines and stuff like that helps them to stay stable in this hydrophobic environment. So different peptides from different places in a protein are going to differ in their hydrophobic profiles. So we can use that to separate them in time such that the least hydrophobic peptides come off the column first and the most hydrophobic peptides come off the column last. Then we give them a good jolt. At this point you put several thousand volts on them and they produce this cloud of ions that can then pass into the mass spectrometer. So electrospray ionization was created by a guy named John Fenn several years ago. He was one of the folks who won the Nobel Prize for creating this science. All right, so electrospray ionization creates a cloud of these ions that are then drawn into the mass spec. Everything else on this bottom row happens inside the mass spec. So inside the mass spec, we're going to get a high-resolution measurement of the peptide ions themselves. All right? So if you have a peptide that is about 10 amino acids in length, if you assume an average amino acid mass of about 110, you would expect a 10 residue peptide to have a mass of 1100 Daltons. Everyone follows that? And if it's carrying 2 protons on board, that 1100 will divide by 2 to give you a mass to charge value of 550. Did everyone follow my math? Okay, so the longer a peptide it is and the types of side chains that it contains will increase its mass, but the more protons it carries, the more that mass will be divided to get you down to a lower m over z value. So a larger mass may be carrying more protons. So there's these 2 countervailing effects. All right, so in high-resolution mass spectrometry, we're going to measure the mass of the mass to charge values of these peptides very, very precisely. Here I've drawn an Orbitrap for this measurement, but it could be, say, a time of flight measurement as well. Now, after we've done this, after we know what peptide masses are present in the mixture, we can set up the instrument to isolate the ions that are intense in that mass spectrum. If you see a great peptide signal over here, you say, aha, we're going to get a tandem mass spectrum of that bad boy. So we isolate a whole bunch of ions at that particular mass to charge value. We cause them to dissociate by bouncing them off of a bunch of gas molecules, and that produces fragments. The fragments of a particular peptide are then collected in a single tandem mass spectrum. All right, that's a lot to take in all at once, and we haven't even gotten to the algorithms, but it's important, I think, for everybody to understand where these tandem mass spectra come from in order to understand what we're going to do with them. Okay. So, now, there are several steps that we've talked about all in a row, and I realize that they might seem a bit of a jumble. Now, we break things apart, we split things, we do all kinds of nonsense, and sometimes it gets confusing to people. So I want to try to focus on 3 ways that we break up proteins en route to tandem mass spectrometry. So denaturation and reduction. Proteins have 3D structure, and that 3D structure is very important to how they function. So one of the things that we need to think about is how we're going to disrupt that structure. So denaturing a protein can be something as simple as dumping it in a bunch of urea, and the thing is going to try to stretch out. Okay? Now, the other piece of it is reduction. What is the reduction step targeting in helping us break up these protein structures? Reduction. Okay. I tend to nickname these the safety pins of the protein structure. Part of the structure of a protein is quite frequently the creation of disulfide bonds between cysteines. Does everyone remember that from protein structure class? Right, so disulfide bonds forming between cysteines hold this structure in place. So you need to break those bonds somehow. We do so by reducing them. One of the compounds you hear about frequently in proteomics is called DTT, the dithiothreatol. Anyone ever work with DTT? Well, maybe someone has, I don't know. But in any case, denaturation and reduction are going to help us eliminate the 3D structure of the protein and get it to stretch out. Once it is stretched out, we can use an enzyme on it. And that enzyme is going to digest particular peptide bonds. The enzyme that we use more than any other is trypsin. Where does trypsin cut? I heard one.
Arginine and lysine.
Arginine and lysine, thank you. Does it say it right there? It doesn't. Good, you guys got it right up. You just knew that. That's brilliant. Okay, yes, trypsin cuts at arginine and lysine. Arginine and lysine are very special amino acids. They're very, very basic in their nature. Arginine is crazy basic. Lysine's pretty basic. So the lysine will cut after these. That's a very important topic because it makes for spectra that are makes for peptides that are about the right length. We'll talk about that in a bit. All right, finally, the only one of these 3 steps that takes place inside the mass spectrometer is collision-induced dissociation. We frequently call it CID for obvious reasons, right? So essentially, if you can cause this peptide to absorb a bunch of this energy, it dissipates it by breaking peptide bonds. And this collision-induced dissociation process produces fragments from the peptide ions that are inside the mass spec. Okay. How is a mass spectrum different from a tandem mass spectrum? I have to say, this has been one of my favorite quiz questions over time. I decided it was so important I would just make a slide about it. So first off, we have a mass spectrum. On the x-axis, we have the mass to charge axis, right? So ions that have large m/z values are at the right, ions that have small m/z values are at the left. Next, we have an intensity axis. That intensity is a very weak proxy for abundance. So never stress that. Just because one ion produces a very intense signal and another one produces a very small signal does not necessarily mean you have more of one than of the other. Because ionizability can be very different among different peptide sequences. So a mass spectrum, each of these spikes represents some m/z at which we have a peptide, probably. When we have a big spike there, we say, we have a peptide that we can clearly work with at this m/z value. That's different than how you interpret a tandem mass spectrum. In a tandem mass spectrum, you're looking at the fragments that result when you blow up one of those. All right? So if you isolate ions at this m/z, all the other peptides are gone. There's just one peptide in the track. Then you cause it to undergo CID, and now all of these fragments are produced from what was originally a single m/z. I'm going to point out one other thing that's really squirrely about mass spectrometry. Remember that the charge of the ion also makes a difference. So if you grab something at m/z 735 and you break it apart, you may see ions with masses like 1200 m/z. So how could you break apart something at 735 m/z and get an ion that's got a higher m/z value? Did we just create mass? We did not. What we did was break apart charge. So imagine that you've got a peptide that's carrying around 2 protons quite happily. If you break a bond, it may be that one proton goes with either side. And so its mass to charge value increases because its charge has decreased. Everyone get that? It's a little counterintuitive, but all right, so a tandem mass spectrum is the death cry of a peptide. Here we see all the little pieces that resulted when we blew this apart. All right, now, how does that reveal sequence? Now we have a 10 amino acid peptide up here, TSIIGTIGPK. What did I say about trypsin? What did you tell me about where it cuts? Arginine and lysine. At the C-terminus of this peptide, we have a?
Lysine.
Lysine, very nice, yes. So don't be confused by the K. K is lysine. What is L?
Leucine.
Leucine, lysine, completely different in their behavior. So here we got our lysine on the C-terminus of a peptide. We are going to break one peptide bond. We're going to break 1, 2, 3, 4. The 4th peptide bond in TSIIGTI. You can see the red line there. It's between the I and the G. We're going to snap that peptide bond through collision-induced dissociation. When we break at a peptide bond, we call the part on the left, on the N-terminal side, a B ion. And we call the part on the right a Y ion. Now you might be asking, well, why would you call them that? That doesn't make any sense whatsoever. So I would note that there are also A ions and C ions and X ions and Z ions, but they are not produced in any amount by collision-induced dissociation. So we're going to just talk about B ions and Y ions. Now you'll note in the red text here that I've specified that the ion at the left is a B4 ion. We call it a B4 because it contains 1, 2, 3, 4 amino acids. It contains the T, the S, the I, and the I. So logically, we call the other ion an after ion. No, I'm just kidding. Before and after, it's fine. We've got 6 amino acids in it. It's the Y ion of the 2, so we call it the Y6. So we started with a 10-mer, 10 amino acids. 4 plus 6 is 10. And here in the tandem mass spectrum, we see the B4 ion shows up at 415.2. and we see that the Y6 appears at 572. Now, how did those masses come about? Well, we know what the mass of threonine and serine and isoleucine are. If not, you can just go look them up in a table. I won't quiz you about it right now. If you add those up and then add one ionizing proton, you get 415. The mass of G plus T plus I plus G plus P plus K plus water plus an ionizing proton gives you this 572. So you might think then, so the sequence is revealed in the tandem mass spectrum. In fact, if you were to cut one amino acid that way, you get a B3Y7 pair, and its mass would be 113 different, because the mass of isoleucine is 113. All right, so the tandem mass spectrum gives evidence about the sequence. Would you feel comfortable reading the sequence from the tandem mass spectrum? It's hard to do. I took a semester-long class devoted to teaching us to interpreting these tandem mass spectra, and I would say it is difficult to interpret and to infer an entire peptide sequence from almost any tandem mass spectrum, even the very beautiful ones that we produce now with high-resolution measurement. So, while this can give us good clues about the sequence, it is hard to just read a sequence from one. All right, so what I want to reveal to you here is that the steps you take on the bench are echoed in reverse by the algorithms. So down the left side, you started with a mixture of proteins, you digested it to produce peptides, you ran it through tandem mass spectrometry to get a bunch of spectra. Now the software is going to have to go the opposite route. It's going to have to reassociate these spectra with the sequences that produce them. From there, we're going to have to decide which peptides we actually trust from that collection. And finally, we will infer a set of proteins based on that set of peptides. So that is a little confusing. What I'm saying is that the measurement, the unit of measurement in tandem mass spectrometry, most typically reveals the identity of a peptide, not of a protein. So once we see the set of peptides we've got, then we can start answering questions about which proteins explain that evidence. All right, this diagram is in many ways, in my thought, the most important one of the whole talk, because this is the bread and butter of proteome informatics. This is a diagram of how database search engines work. Database search engines are like the central tool. They are the tractor of our fields. And everyone who does proteomics at some point is going to use a database search engine. So, how do they work? The creator of the engines once wrote that database search engines look up the answer in the back of the book. And that is a little unfair, but we'll just start with that. We start with the notion that the samples we analyze are not from some alien species we've never encountered before. They're from species that we typically have a great wealth of genomic evidence from. So if you're working with a human sample, you can leverage the human genome to help you interpret the spectra. Because if you started with a human sample, the peptides in that sample should largely come from humans. So we start with a FASTA sequence database. FASTA is an old sequence alignment algorithms, but one of its greatest gifts to the field of bioinformatics was a very standard way, a very standard text file format for reporting bunches and bunches of protein and nucleotide sequences. So we start with a FASTA database that contains all of the protein sequences known for this particular species. Where can you get these? Anywhere. You can get them from Ensemble, from Uniprot, from RefSeq, lots and lots of places. So getting a FASTA sequence database is effectively giving us the universe of all sequences we're going to consider. And an important corollary, if the protein you're looking for is not in the database, you can't identify it. Let's imagine that you are an HIV researcher and you're trying to identify the proteins that come out of CD4 cells. So when you have these cells, if you run a search of their proteins, including only the human proteome, there is no possibility whatsoever that you would detect any peptides from HIV itself. You must include the sequences in the database in order to see them. All right, so our universe of the possible protein sequences comes from this first step. Next, In silico, which means in the software, the algorithm is going to attempt to emulate the behavior of trypsin. So trypsin on the bench, run overnight, is going to digest after lysines and arginines. Thank you for that. In silico, we must emulate what peptides should result from protein sequences if trypsin were to act upon them. All straightforward, hopefully. So that gives us our list of peptide sequences. Next, it may be that you as a researcher are very interested in the post-translational modifications of the sample. That's something you can measure with proteomics, but could never measure with microarrays or with sequencing, right? We have to look at the proteins themselves for that. So maybe you've enriched for phosphopeptides, therefore you must tell your search engine, I'm interested in phosphopeptides. And if so, the PTM expander will allow for that possibility. I call these decorated peptides. Now, is it true that every spectrum must be compared to every peptide from the human proteome? We got a head shaking no, and that's absolutely correct. Some peptides are comparable to a given spectrum. Some peptides are not. And we make that judgment based on the fact that we have a highly accurate mass of this peptide before we blew it apart. So if something is very close to that observed peptide mass, or mass to charge, we will examine the fragments that should result from it and compare it. If it's very far away, 50 Daltons away, there's no point whatsoever in our comparing this peptide sequence to a given spectrum. Okay, so the peptide mass filter has been one of the ways that the database search engine prevents us from taking years and years to try to analyze some sample, because only those peptides that are very close in mass to the observed peptide need to be compared. So now we have a list of candidate peptides that should be compared to this particular spectrum. At long last, we're finally going to make use of the fragment information that we collected in the Tandem Mass spectrum. So from a given peptide, we're going to predict which fragment ions should result, and then score the set of fragments that we predicted for this peptide versus those that we actually saw in the spectrum. Now you might be asking, well gosh, how much did we limit our search space? You know, we had 100,000 proteins in our human database, and those produced, let's say, 10 peptides per protein. So how many peptides did we really end up comparing to each spectrum? That answer depends a lot on how you set up the search. So we're going to go into some of the determinants of that in a bit, but I would say that in general, you're going to have more than 1,000 possible peptides that could explain the presence of each spectrum. Some of them are going to be a close fit, and some of them are not. All right, so let's start with the peptide mass filter. Peptides can only be compared with the tandem mass spectrum if their computed masses, the weights that we get for these peptides by adding together their residues, if they agree with the measured mass within some precursor mass tolerance. What precursor mass tolerance you use is going to depend very heavily on what kind of instrument you use to measure it. So maybe you used an old LTQ instrument. This is a linear ion trap, very venerable instrument now. If so, the instrument maybe gives you a mass measurement for the peptide that's as far away as 1.25 m/z. That's a pretty big range of error for that kind of instrument, but this is the reality of it. These instruments are not known for high mass accuracy. Maybe you used a time of flight instrument. Anyone have a notion what kind of instrument you have in the proteomics lab here? It's called a TOF, which is a time of flight instrument. So its measurements are easily within 100 parts per million mass accuracy. That's much, much better than any ion trap is ever going to produce. So that's very highly mass accurate. This 100 ppm is actually very wide for an instrument like that. Yours would probably be easily accurate within 50 ppm of any observed ion. Now maybe you're using one of the newfangled Orbitrap or FT class instruments. So we have some of these over at places like CPGR, and of course at our campus and over at UCT. So Orbitraps are pretty easily found in this country. So that means that you may know the mass of the precursor ion within 10 ppm. So that's a very vanishingly small, maybe within 0.002 m over z. It's a tiny, tiny window of accuracy. So the accuracy of the instrument is then going to affect how many candidate sequences must be compared to each spectrum. Okay, so let's get down to the nitty-gritty. What actually differentiates database search engines? Now we're starting with one that was published back in 1994. Was anyone in this room born before 1994? Okay, I graduated high school by that time, so anyway. So back in 1994, a guy named Jimmy Eng, who was a mechanical engineer who was hired by John Yates, created some software that would automatically determine the identity of a peptide associated with a tandem mass spectrum. That software was called Sequest. A lot of people confused it for a popular show from way back in time called Seaquest, which was set at sea and involved looking at marine animals. But that's okay. Sequest quickly became a very standard tool for our field. In fact, a company that sold mass spectrometers called Thermo Finnegan decided to sell licenses to Sequest with their instruments and give some of that money back to the lab. So this was actually a money stream for my lab, for the lab where I got my PhD for some time. So his notion was that we don't know very well what kind of ions should be produced by breaking a peptide. So maybe we should make the spectra we observe look a little bit more like what we can model peptides to look like. So they normalize the spectrum. They chop it up into 10 zones, and each region, all the peak intensities are normalized so that the highest peak intensity is 1. Next, generate a model spectrum for each candidate. This was a fairly accurate, actually, initial attempt to figure out what a spectrum should look like for a given peptide. The software says, what fragment ion would be produced if we broke after the first amino acid, and the second amino acid, and the third amino acid, and the fourth amino acid. And in each case, it said, if the fragment contains the C-terminus, if it's the Y ion, we'll model it as a height of 2, and if it contains the N-terminus, we'll give it a height of 1. So this is a very simple kind of model for what the spectrum should look like, but after normalizing it, normalizing the spectrum we collected, it actually is not a bad projection of what these tandem mass spectra should look like. Then, it uses a technique called fast Fourier transform. Fast Fourier transform is beyond the purposes of this class for now, but I will just say that it is a very useful way, a very useful shortcut to get to cross-correlation. Now I'm going to try to explain this. Imagine that this hand represents the observed tandem mass spectrum, right? We've got a low of, say, 0, and here we've got a high of 2,000, and here we've got a bunch of peaks that represent observed tandem mass spectral peaks. These are fragment ions. Now, I have another spectrum. This is one that I've predicted on the basis of some peptide sequence in the database. So I want you to imagine that if I line them up with what we call 0 offset, that the observed ion for 385.2 aligns with a fragment ion that I've modeled to be at 385.2. So I can compute a cross-correlation by asking how much do they continue to align if I scoot one relative to the other. You know, at some point even like the peak at 500 in one aligns with the peak at 380 in the other. This helps us to evaluate kind of a background matching. So we run it back and forth across itself. We produce a data structure that looks like this. So here we have this spike at the middle that represents how well they line up when they're aligned with no offset, and then all this noise represents all the junk alignment that happens when they're spaced out from each other. We ask how much better is this spike than the noise surrounding it, and that's the score that we associate with this peptide. And it's called the X-score. So the X-score score has been very, very robust. This software has been in use since before everyone in this room was born, except for me. And those scores are still in use today in software that still bears the name Sequest or Sequest HT, part of Proteome Discoverer now. So X-score was a really old idea borrowed from engineering techniques, but it's turned out to be highly useful in this context. I just want to throw in one note about the second citation I put in here. There's another paper from 2008, also by Jimmy Eng, and here Jimmy says, you know guys, I messed up. Back in 1994, when I published this first scorer, I didn't realize that there was a simplification I could do in my math that makes Sequest run 10 times faster. So people had burned CPUs cursing how long it took to get their search results for a decade, and then finally, oh look, here's a way to do it faster. Awkward, but true. So sometimes the math gets better with time. Now, Sequest is far from the only tool people use for this purpose. One of the biggies that you see a lot of use of, for example, at University of Cape Town, is MaxQuant. MaxQuant came out of a guy named Jürgen Cox working in Matthias Mann's lab a while back. So MaxQuant has its very own search engine built in called Andromeda. Everyone heard of Andromeda? Anyone know what that refers to? It's another galaxy. Sorry. So here we go. We have the Andromeda search engine. Their approach is quite different. Now, before, we were talking about one spectrum sliding past the other and asking how much better they were when aligned versus when they were not. Andromeda is something that we call a match counter. There are lots of algorithms that use techniques like this, so I thought it was a good example to use for that purpose. So we're using the binomial distribution. This is one of a family of distributions we use in statistics for asking things like how many coin flips do you need in order to accumulate 38 heads, for example? That sort of thing. So the binomial distribution is very good for asking how many matches would you have between a peptide sequence and this spectrum by random chance. And it answers that through this math. This is a combination. This is the number of peaks that appear per 100 m/z units in the tandem mass spectrum. And this reflects the probability of not getting such a match. So I won't go through all the math right now, but you can see that it has this summation right here. That is to say, what is the probability that you matched 38 if you match 38 fragment ions, but also the probability that you match 39 and 40 and 41 and so on. So it's looping upwards until it hits the maximum number of matches possible. That's how it gets this thing that we would think of as a p-value. What's the probability that by random chance you would match this many or more fragments? It also does a little bit of optimization, and these are kind of not really statistical perspectives, but they're an additional spin on this. So the first is that the software realizes that certain kinds of post-translational modifications that we look for, like phosphorylation on serine and threonine, can lead to extra fragments showing up. So the software scores such peptides two ways. One in a case where the loss doesn't take place, and one in a case where it does. The other thing that it does, and this is sort of a squirrely thing that it shares, that it has in common with an algorithm called Mascot that's been around for forever. It optimizes how many peaks are called peaks. That might seem a little strange, right? If you observe something in a tandem mass spectrum, you might think, well, obviously it's been observed, therefore it's real. But in fact, there is some amount of chemical noise and some amount of physical noise that we have in these spectra. So the software says, look, if I say nothing below this line will be counted as a peak, does my match get better or worse? So by adjusting which peaks get counted as signal and which ones get counted as noise, the software optimizes for the best possible score for each peptide sequence. So those are some of the secrets buried under the hood in Andromeda. This is right out of the paper that they published on it. Okay, now there's another algorithm that's kind of funky, and I feel like explaining its innards is going to be a little challenging. But I would point out that it's been, in our hands, has shown itself to be very, very powerful for discriminating good matches from bad, and I wanted to talk about it. So it's called MSGF Plus. I always feel like I need to have a little conversation with them about coming up with intuitive-sounding names, right? But that's okay. MSGF Plus. It's named because it is a mass spectrometry generating function. Whoa. So the generating function comes in at a later step. Let's start by the fact that MSGF Plus is not like other database search engines in that it does not score against the spectrum as it is. Instead, they transform the spectra in a way that tries to say, what is the probability that there was a breakage in this peptide in mass units from the N-terminal end? Isn't that squirrely? The math behind it is a little confusing, but let's just say that if you see a B4 and a Y6 ion that added together give you the mass of the whole peptide, they're correlated with each other, right? Because one breakage produces both of those fragments. So their software does a transform of the spectrum in a way that tries to bring this information back together before it does match scoring. And then the generating function itself does something that sounds on its face rather impossible. It asks, what is the probability that a match this good or better how many matches this good or better would we expect if we compared against the space of all possible sequences? Well, that's kind of weird, isn't it? Because in a normal database search engine, you're only searching the sequences that are in the database. And so if you get a small database or a big database, you're going to have to evaluate the result based on how many comparisons you did. But in MSGF Plus, you're comparing the score that you get from this to all possible peptides and the scores that they would produce. It sounds impossible, but the expectation value that they report has been shown to work really well and to be very stable against different database search sizes. And in later versions, they even made the software work really brilliantly when you have high-resolution measurement of fragment ions, and that has become a very popular kind of data to produce in tandem mass spectrometry. So this was published back in 2014. So over time, more than 30 database search engines have passed into peer-reviewed literature. So it's become a very popular thing. Almost everyone entering my field, the field of proteome bioinformatics, would introduce themselves by releasing yet another database search engine. So you have a lot of choice now in which one you choose. All right, now what have we accomplished by this? All these scores that I'm talking about really have two very different purposes. And this is something I want everybody to understand. When we do a database search on an individual tandem mass spectrum, it's very important that the correct sequence get boosted all the way to the top. I think everyone can see that. You've got 1,000 or 10,000 or 100,000 different sequences all being compared to this spectrum. At base, if the correct sequence doesn't make it all the way to the top of that list by its score, we're not going to use it. So that's a pretty hard criterion. For every one of these spectra, we want the best sequence to win, and ideally that best sequence would actually be correct. So that is the role that everyone thinks of with these database search tools, that the best sequence, the correct sequence ideally, will be boosted all the way to the top. And if you're really going to be sensitive about it, even pretty marginal spectra need to be able to get their correct sequences boosted all the way to the top. The spectra are not all gorgeous. The ones that get the best scores are, but the ones that gain you sensitivity are generally kind of bad looking. The other problem is that we need to figure out which spectra have acceptable identifications. Some of the spectra are simply not going to be identified. They may be very low signal to noise. They may represent polymers that are running through the column. So some number of these spectra are going to fail in the identification process. Even a relatively bad instrument these days can generate 10,000 tandem mass spectra in an hour-long experiment. 10,000 tandem mass spectra, right? You would like to be able to say, from these 10,000 spectra, I got 10,000 peptides. But the reality is that if you identify 50% of the spectra you collect in a proteomics experiment, you're doing really well. Half the data being identified constitutes brilliant success. Generally, people don't complain about the result of their database search until they get down to something like only 10% of the spectra identified successfully, right? So people start with this relatively low bar, that of all the data collected in an experiment, if they can identify just half of it, they feel pretty good about it. So these search scores are not just to be used to figure out which sequence wins for each spectrum. It also plays a role in our deciding which spectra we are going to call successfully identified. So sometimes you'll see the term well-calibrated used for this, meaning that the scores produced for individual spectra are also comparable among spectra. This is one of the features that people really like about MSGF+, that the scores can be compared among spectra because they're all considered against the full population of all sequences. Okay. So, how do we control PSM error? PSM is a term that gets thrown around a lot, so I want to make sure that I help define it for you. A PSM is a peptide spectrum match. I have some spectrum. My database search engine ranked this sequence first for it. And so I have this PSM that represents this connection between this spectrum and this best sequence from my search. Now I need to decide, is this PSM acceptable or am I going to throw it out? So for this, we're going to start by looking at two different methods. Now, one of these methods is fairly computationally complex. The other one is fairly computationally simple. And you might think, well, gosh, I guess we just have to use the simple method then. But the reality is that there are times when you need to know the accuracy of individual PSMs. Let me give an obvious example for this. Maybe before you join a lab for a master's degree, your principal investigator has run a proteomics experiment. They now have 3 spectra that match to a protein that got them very, very excited. And they want your 2 years as a master's student to be invested in figuring out what this protein does. Do you want to have the next 2 years of your life built around whether this protein is real or not? This is difficult, right? So sometimes people make investments of serious amounts of time and money and effort in trying to chase results that really shouldn't be chased. So having accuracy, having accurate identification data built from proteomes really, really does matter. So we're going to talk about distributional modeling that asks how the scores distribute for false PSMs and for true PSMs. And from that, we'll try to assign a probability to each PSM to say how accurate we think this is. This one has an 80% chance of accuracy. This one has a 95% chance of accuracy, that sort of resolution. And then we get to the target-decoy method. And the goals of target-decoy are very, very different. In target-decoy, we're simply trying to say, out of this collection of IDs that we have accepted, what fraction are bogus? Everyone sees that? So we don't have the ability to say, for this individual PSM, what is its accuracy from target-decoy. We can, however, make a statement about the collective of all of the IDs we've accepted. So distributional modeling is a very fine-tuned method that gives us point information about individual PSMs. Target-decoy gives us an estimate of the total collection. All right. So this, the approach for distributional modeling is actually a pretty old one now. It was published back in 2002 by Keller and Nesvizhky out of Eberswald lab. So what they did was to look at a whole bunch of PSMs from, I believe it was H. influenza, just a bug basically. In searching their data, they included the H. flu database, all the proteins known to be from this particular bug, and they also included a whole bunch of proteins that were not in this sample. So H. flu, and then a whole bunch of distractors. And then they said, for every spectrum that matched best to H. flu out of this very large database, we're going to say is a true sequence. Every spectrum that matched to something else, we're going to call a false sequence. And then they looked at the distributions of how their scores compared. And you see that we have these low scores that had this huge peak, and then this very small number of spectra that had positive, that were for the true species. This represented, by the way, one of these older data sets where fewer than 10% of the spectra were identified. This is off an old LCQ instrument, way back in the ion trap days. So you can see that for a given discriminant score that can be generated for a given PSM, if it's way over here, you're going to have a lot more confidence in it than if it's way down here. Does everyone see that? Because if it's down here, there's almost no possibility it comes from this distribution, and quite a large possibility that it comes from the negative distribution. If something falls right at this line, say a little bit south of 2 on discriminant score, it's got roughly an equal chance of being from the positive distribution or from the negative distribution. So this is why, for an individual PSM, you can put some probability of correctness on it. Now, a lot of people find that very hard to deal with. So I'm going to talk about the target-decoy method because an awful lot of people make use of this. And there are some ways that you can game the system so that you get false results out of target-decoy. But in general, when people are just using it for protein ID stuff, they come up with pretty sensible numbers. So I want you to imagine that I have sorted all of the spectra from this file beneath my hand. This one at the top got a really great score. This one down on the floor is obviously palpably false, right? Now I need to figure out where I'm going to set a threshold against this list. If I set it way up here, I'm safe. All of the IDs above my threshold are perfectly solid. If I set it way down here, my sensitivity got better, right? I saw more peptides that were real, but I also erroneously accepted a lot of things that were false. So we need to find where to set this line so that we maximize our sensitivity without blowing apart specificity. So in this case, for a given place where we set the threshold, we ask, well, we're going to start by searching in our database against a collection of peptide sequences, some of which are real and some of which we know are false. So you remember just a moment ago we were talking about the H-flu experiment that they did with the distribution modeling. In that case, they had a relatively small number of sequences in their database that were true and a huge number of sequences in their database that were false. In target-decoy, we're going to even that up. So if I'm searching a human database, I'll have my 100,000 proteins that are true, and then I'll include each of those protein sequences reversed, end for end. Now, every time I match a spectrum to these reversed sequence entries, I know it's bogus. I know it's completely false. Anytime I match a spectrum to the normal orientation entries, though, it could be true or it could be false. I don't know it's true just because it hits a true human protein. So when I look at a particular threshold on this list, the IDs that are above it, some of them are going to be from from real sequences, and some of them are going to be from bogus sequences. So my assumption is this: for every bogus PSM above this list, obviously bogus one, there's another one hiding among the possibly true sequences. So when I look at a particular threshold, I can double the number of decoys that are above my threshold and say that's the number of false entries on my list, and divide the total number that are above this above my arm as the total number. So if I keep that number under, say, 5%, I probably have a set of IDs that are about 95% accuracy. Of which 95% of them are actually true. So this target-decoy method is quite simple, and yet it has the limitation that it can only give you a collective assessment of the IDs. It doesn't tell you whether any individual ID is right or not. All right, this brings us to the final part of this. I know some of us are wearing down, but if you'll stay with me for another 10 minutes, 15 minutes, we're going to make it. We're going to make it through. All right, so how do we get to proteins? Biologists doing proteomics in general don't care about peptides. What they care about is your protein list. So how do we get to that? First off, why is this even difficult? Why would it be that peptides are shared among different proteins? So Kunin wrote this really useful definition a while back in a paper from 2001. Orthologs are direct evolutionary counterparts derived from a common ancestor through vertical descent. Whenever we speak of the same gene in different species, we actually mean orthologs. Okay, so we're humans, right? Humans have a very special evolutionary history. And yet, if you look at the protein sequences we possess, and the protein sequences that chimpanzees possess, you'll see an amazing degree of similarity between the two. If you look at the genes you possess and the genes that a yeast possesses, you will still find points of similarity. So orthology is something we must think about if the sequence databases we use contain proteins of multiple species. So if you were to use SwissProt as your search space, SwissProt has protein sequences from hundreds, or I think even more than 1,000 species in it at this point. So if you identify a peptide from actin in a human sample, such a database would also say, well, it's possible that this actin came from chimpanzees or from bonobos or whatever. So being able to deal with situations like that matters. Being able to deal with orthologs, where we have the same protein occurring in multiple species, matters. In contrast, paralogs are genes within the same genome that have evolved by duplication. Now, I know that some of you work with agricultural species, and one of the less pleasant features of certain plants, particularly trees, is this degree of gene duplication that they go through. You may find a hexaploidy in some cases. In cases like this, we have a great number of paralogs where the same gene appears in multiple places in the genome with slightly different sequences in each. And these paralogs can overlap very significantly in which peptides they contain. So, I want to introduce the term parsimony to you. Now, if we can take a brief moment, just a quick quiz break. Who here has an older brother or sister? Older brother or sister? We have a few. Okay. Would anyone care to share a name that you were called by an older brother or older sister? Yeah, only polite terms. Oh, no dirty language? Oh. I had an older brother. He was 2 years older than me. His name is Tom. So he may actually come to South Africa for the first time in August. I'm very excited about this. But my brother Tom would frequently be with me as we walked around in a store. We would go to Walmart, and I'd go to the toy section as a child, and I would look at the things and say, gosh, I would like that Saturn V rocket. I would really like that Saturn V rocket. And my brother would say, why don't you just buy it? I'd say, I don't know, I got to save up more money. I got to save up more money. And my brother would say, David, you are so cheap. Cheap. Oh, I don't know how many times I got called cheap in my life. That hurt every time. So I want to ask you, put yourself in the same situation, an older brother, older sister, hanging around near you, and they break one out on you and say, why are you so parsimonious? How do you feel about that? Positive, negative? Parsimonious is very close in meaning to my brother's insult, calling me cheap. And in science, we consider it a good thing to be cheap. In parsimony, we never want our explanation to go further than our data can take us. All right? So when we are naming what set of proteins we've identified from a sample, we don't want to spend more proteins than we must to explain the peptides that we've observed. That is a parsimonious principle. This comes from a fellow named William of Ockham back in the 14th century, so the 1300s, who said something like, plurality, naming lots of things, should never take place, never be posed without necessity. So in other words, don't go further in your explanation than you must in order to explain what actually has been observed. Now, he actually said this in Latin. Did anyone study Latin along the way? I'm just going to wreck every Latin scholar's ear here. Pluralitas non est ponenda sine necessitate. Okay, so this concept from the 14th century still applies in science today. We want to be parsimonious, ensuring that our explanation doesn't go any further than what the data actually support. So, this is a situation that is a real situation. This came from a data set generated by friends of mine at the Broad Institute. Carl Klauser is a great guy. So, he identified all of these yellow peptides at the right, and then we looked at which proteins in the sequence database mapped to them. Now, this was kind of messy because this is called a xenograft. This is a human tumor growing inside a mouse's body. So we had the possibility of having mouse proteins or human proteins to explain the peptides we saw. So here we see that we had two peptides up at the top. Those could be explained by this first protein, the second protein, the third protein, and this one way down here. And you can see I've actually boxed together three proteins that all map to the same set of peptides. So how do you break up situations like this? How do you enumerate the smallest number of proteins possible to explain all of the peptides you have at the right? This is called a bipartite graph, a graph of two different types of things, where all of the edges run from an item of type 1 to an item of type 2. So the question of how do we find the smallest number of proteins to account for all of the peptides is something that we call a set cover, and it's actually a very hard problem to solve. So I'm going to provide a couple hints on how we do this. Let's start with the concept of indiscernibility. we have a large number of equivalent proteins. And we say that they're equivalent because you may have situations where this set of peptides could be explained by the presence of protein A or the presence of protein B, and neither one of them is an advantage in explaining the data. So in our software, we typically say these count as one protein, and either accession would be equally good in making the explanation of why these peptides were present. Does everyone see that from the graph here? So I draw these edges to say protein A could explain the presence of peptide A, peptide 1, protein A could explain the presence of peptide 2, and protein A could explain the presence of peptide 3. The same is true, all three of those statements are true of protein B as well. Therefore, we group them together and say this is one protein, and we're not sure which of these two it is. Equivalent or indiscernible proteins. So that's a simple way to simplify our protein lists. But sometimes we use this parsimony rule to rule out some of these. So imagine a scenario where protein A links to peptides 1, 2, and 3. So simply invoking protein A will explain all of that evidence. But look, protein B also maps to peptides 2 and 3. In proteomics, we will frequently apply a subset rule, or a parsimony rule in this case, to say we're not going to list protein B. Instead, we will list protein A, because only protein A, in this case, can explain the presence of peptide 1. Protein B, we're going to just rule right out, because we've got no specific evidence of its being there. We have more than enough evidence to say A is there, but once we've said A is there, we no longer need B to be there to explain any of the evidence. It's a subset protein. So frequently, software will simply omit protein B. It'll cross it right off the list, and it will never be presented to you as a protein that was present in that sample. Now, what happens if you run this experiment again, and this time you see a peptide 4 that could only be explained by the presence of protein B? Suddenly, it might appear that protein B has lots of evidence in sample 2 and 0 evidence in protein 1. So we have to be very careful about how we do protein inference so that we don't cause proteins to vanish and to spontaneously vanish and appear across different samples. In total, we do quite a lot by applying these indiscernible rules and parsimony rules. In this case, we have a protein sample that contains 49 proteins from humans. We know they're from humans. When we search it against SwissProt, which at the time I think had about 250,000 different protein sequences in it, we got more than 400 proteins identified from these data. 400 proteins identified when only 49 were present. So when we apply the grouping of indiscernibles, that number falls quite a lot, to 150 in this case. But once we apply parsimony on top of that, we get back down to this very small 49 protein list that we do from some of these other human databases that we worked with. So applying parsimony and being very careful about adding to your protein list to account for the peptides you see can do quite a lot to cut down the bulk of proteins to a list that's actually accurate. All right, there were a lot of messages today. I realized this was kind of a long go at proteome informatics, but I hope that everyone is able to take something away from this. We start with the fact that proteomic identification depends upon a database of known protein sequences, usually from genomics projects, that define the set of possible peptides. If it's not in the database, you won't identify it. Next, controlling the error in the identified peptides is absolutely critical. In fact, if you don't apply some sort of method that gives you an estimated false discovery rate, you won't be able to publish in most of the journals that serve the proteomics field. So everyone must be able to control the false discovery rate among their accepted peptides. And finally, detecting indiscernible proteins and applying parsimony helps us have sort of a conservative appraisal of what proteins have resulted in the data we've seen. If you don't use rules like this, your protein list will be very inflated. So you want to make sure that whatever toolkit you're using has some way of minimizing the protein lists to just those that are required to explain the peptides you've seen. Okay, that was pretty whirlwind. but I'm really glad you stuck with me throughout. I'm happy to take some questions here if you have some. But if not, I would ask you to just shoot me an email and I'll be glad to try to help out. I believe next week you're going to have everybody back from North Africa, and I look forward to hearing what they have to tell you about this fine field. But I hope that this introduction to the algorithms is helpful to you. Thank you.